In this video, I want to talk about the specifics of DNA replication. What are the enzymes involved, and what does the replication fork look like? So, what needs to happen in DNA replication? There are three main things that we want to keep in mind. So, the first thing that we need to do is we need to separate the two DNA strands. And we also want to keep them apart as we're polymerizing. We want to keep them apart. Then what we want to do is we want to create or synthesize a new anti-parallel and complementary strand for each existing strand. Meanwhile, we want to make sure nothing gets messed up. Make sure nothing gets messed up. We want to make sure that nothing is damaged or ruined when we're doing all of this. So what's going to happen? So we start off with double-stranded DNA, and then this enzyme, I'm going to draw it as like a green circle here, something like this, comes over and it begins to unwind the helix. This thing is called helicase. So helicase comes in and it melts or unzips the DNA. And then we get the little replication bubble. So we're going to zoom in on this portion here. This portion is called the replication fork. Okay. So now if we imagine we have a strand, this top strand is running 5' to 3', and that means this bottom strand is running 5' to 3' the other way. So the first thing that happens is, of course, this helicase is working on this, trying to go through and unwind the helix some more. Okay, it's pulling these strands apart. This is helicase. So while helicase is doing that, I actually haven't drawn it here. I've just drawn it as two straight lines, but keep in mind that this DNA strand would actually be a double helix, right? It would be twisted about itself. So when helicase comes in here and it pulls these strands apart, what it causes is the turns here of the double helix to be more tightly coiled. So helicase creates supercoils. I want to mention that. It creates supercoils, which are basically just really tight coils. So there's this enzyme called topoisomerase, and what topoisomerase does is it relieves the tension that these supercoils cause. So this here I'm going to draw as topoisomerase. This relieves tension caused by supercoils. Okay. There is a specific topoisomerase called DNA gyrase that exists in prokaryotes. Okay, DNA gyrase is a specific topoisomerase, and the way it works is it actually adds negative supercoils to positive supercoils to undo them. Okay, so topoisomerase is the general term associated with an enzyme that relieves the tension caused by supercoils, and DNA gyrase is a specific one that exists in prokaryotes. Okay, so helicase is unwinding this helix. As soon as these strands here are free, then there are these little proteins that bind these strands, these single-stranded DNA. So this here is still double-stranded DNA, but these here are single strands. Now these single strands are bound by these little proteins. These things are called single-stranded binding proteins, right, or SSBP. What they do, they actually have two functions. One thing is that they protect the single-stranded DNA from degradation. Okay, so these strands, once they're single-stranded, they can be acted upon by nucleases maybe in the environment. So these single-stranded binding proteins protect the DNA strand from being degraded. Another thing is it also keeps these strands from reannealing. So if you imagine it kind of like this, it keeps the strands from coming back together. Keeps the single-stranded DNA molecules apart. Okay. So, what's going to happen after that? Then our good old friend that we've mentioned before, primase, comes in and lays down an RNA primer. Okay, so now let's think about this for a second. If this is the 3' end here, that means the new DNA needs to be running starting from here, from the 5' going down this way, right? And if this is the 5' end of this one, then the DNA here is going to be going towards it, right? 5' to 3'. So we're going to add a primer right here, and that's going to be the 5' end. Okay, so this primer here, that is a primer, and that is laid down by primase, right, which is a special RNA polymerase, because primase can synthesize de novo. So once that is there, what's the purpose of that? The purpose of that is to have, or to give, DNA polymerase a free 3' OH group to act on. So once this primer is here, then DNA polymerase can go in and begin to polymerize 5' to 3'. So it'll start going this way. Okay, so that blue portion, that DNA, is going to be done by DNA polymerase. Now, if this is a eukaryote, it would just be a DNA polymerase. But in a prokaryote, it would be DNA polymerase 3 in prokaryotes, because it's the main DNA polymerase as far as extending the chain. So DNA polymerase and DNA polymerase 3 in prokaryotes. This will elongate the DNA. Now, of course, it's doing it 5' to 3', right? Starting at the 5' end, going towards the 3' end. Now, this one gets to go down like this. So now what you have to bear in mind is that helicase is going to continue unwinding these two strands. So when it does that, that'll allow this DNA polymerase to continue to go 5' to 3'. But notice this bottom strand is going to have a problem, because this is the 5' end, which means the 3' end will end over here. So we have to start 5'. So the primer, primase is going to lay down a primer. It can lay it down. The best place to lay it down is right here, right? Because that's the furthest place that it can add it. So that's going to be the 5' end there, and then DNA polymerase can come in and extend it 5' towards the 3'. Okay, so that occurs there. So primer lays down both of these, or primase lays down both of these primers here, right? And then DNA polymerase extends both of those chains. Okay. Now, moments later, what we'll see is that this strand up here can continually synthesize. Right, if we have that primer up there, it can continue to synthesize as this thing opens up some more. Okay, I'm not going to draw helicase or topoisomerase just for the sake of time. I don't want to make this video take forever. But this thing can keep going. It can synthesize continuously. But this bottom strand, we initially had this primer, right? So maybe that was like right here somewhere, and then from there we went down and went ahead and went 5' to 3'. So here at the top, here we went 5' to 3', and we're still going strong, 5' to 3' in this direction. This one went 5' to 3', and then it's done, right? But it hasn't synthesized this portion. So now another primer needs to be added here, because now that the strand has opened up some more, another primer can be laid down, and then extension of the DNA can occur some more. So now this top strand has a specific name. It's called the leading strand. Oops, that's really thick. Let me make that thinner a little bit. This top strand is called the leading strand because it synthesizes continuously. This bottom strand, however, is called the lagging strand because it synthesizes discontinuously. Oops, not discontinuously. Because as it opens up more and more, we can lay down another primer, and then it can elongate. Lay down another primer, it can elongate. So these little portions here are called, as you may know, Okazaki fragments. So this here is an Okazaki fragment. This here is an Okazaki fragment. These are Okazaki fragments. These are Okazaki fragments. So these Okazaki fragments, they have these little gaps between them. So those little gaps need to be sealed. So what seals them? Well, there's an enzyme called DNA ligase. It'll go through and seal those gaps. Okay. So these Okazaki fragments, a little bit more information about them is that they'll exist both in prokaryotes and eukaryotes, but in prokaryotes they will be about 1,000 to 2,000 nucleotides long. NT stands for nucleotides, whereas eukaryotes, they'll be shorter. They'll be around 100 to 200 nucleotides long, which is kind of a little bit counterintuitive. You would think that they would be, because the chromosomes of eukaryotes are longer, you would think these Okazaki fragments would be longer, but that's not the case. They're actually shorter. Okay, so eventually this will complete, and you will have two double-stranded DNA molecules coming from one. Okay, this is of course a semi-conservative replication, right? These two gray strands of the initial parent strands, but eventually what you'll get is two strands that are with one gray strand and one blue strand. So one thing I do want to mention though is that the enzyme DNA polymerase 1, in the case of prokaryotes, right? This is assuming it's a prokaryote. We'll go back, get rid of that primer. It'll remove the primer and replace it with DNA. In eukaryotes, it's a little bit different. There's this enzyme called LEN, which is a certain nuclease, and there's also this thing called RNase H, which is involved in removing the primer. Okay, those things remove the primer in eukaryotes, and we'll talk about that some more later. Okay, so that's pretty much it as far as the enzymes go for DNA replication. Hope that video was helpful. One last thing. I am a tutor. If you live in Southern California, feel free to contact me via email at moofuniversity@gmail.com and see the description below for more details. Thank you for watching.